Okay, so when we last talked, we saw examples here of an error here where we had seals and whales grouped together. And if you look here, we have another error shown up here where we have deer and hippopotamus lumped together when they shouldn't be. In this case, a paraphyletic group, and down here, a polyphyletic group. Here we leave out the common ancestor. Here we down here we leave out the common ancestor. Here we include the common ancestor, but we don't include all descendants of that common ancestor. So there's two different errors, and these are different ways that we've messed up classifications in the past. Now, how do we make these classifications to see if we've created errors? Well, we start by looking for ancestral and derived characteristics that are shared. So what we're talking about are homologous traits here. Okay, we're looking for homologous traits that are shared or derived. And what do we mean by that? They're ancestral or derived, I should say. A shared ancestral character is a homologous trait that is inherited because it evolved in an ancestor of the taxon. A shared derived character is an evolutionary trait that's unique to that particular clade. So it's derived, meaning it's newer, it's new, it helps separate one group from another. So let me show you an example of that. So let's say this is the fish, this is humans, this is dogs. Okay? Vertebrae show up down here. And everyone on the cladogram has inherited vertebrae from a common ancestor. This is a shared ancestral trait of fish, humans, and dogs. Humans didn't evolve vertebrates separate from dogs or fish. We inherited them from a common ancestor. So it's a shared ancestral character. Okay? A shared derived character would be a trait like, let's just say, fur or hair that separates mammals from fish, that evolved later, that unites all of these organisms together but separates them from the rest. That's a shared derived trait. Okay? Now, a trait can be ancestral and derived depending on how you look at it. For example, going back to our idea, let's just redraw this in a slightly different way. A similar looking tree, but let's just have humans here and fish here, and we'll say this is, I don't know, squid. All right. Vertebrae in this case unite the vertebrates, right? So you could say vertebrae are a derived trait of the vertebrates that unites all the vertebrates, which I'll circle here in yellow, the vertebrates. But within the vertebrate clade is the clade of mammals, which will make kind of a blue color here, and they have a derived trait of fur, which unites the mammals and separates them from the fish. So you could say vertebrae are a derived trait of the vertebrates, but they're an ancestral trait of mammals, of humans in this case, depending on how you want to view or what scale you're looking at it from. Okay. Now, how do we build one of these? So it says how to building a phylogeny. If we want to, let me rephrase this. Let's build a cladogram, because that's what the tree is called. That's the techniques we're using, our cladistics. A cladogram depicting phylogeny. First of all, we find shared ancestral characters of everyone on the tree and shared derived characters of everyone on the tree. If we can, you know, what traits do they have that are unique? What characters do they have that are shared? And are there any unique derived characters that really make different groups different from one another? You know, this one here you can kind of ignore. It's kind of the same as the bullet above. Then we want to determine our outgroup. That's the group that is known to have diverged first before the in-group, which is the group of interest. So when we draw a tree like the one I did before, you know, this is our outgroup over here, the outgroup, and this is the in-group. So what group was one of the first ones to split off? That helps you to orient your tree so you know the order of evolution. Okay. So here's an example. And first of all, we make a character table, or I'll call it often in lab a data matrix. And what you can do is you can use zeros to indicate that a trait is absent and a 1 to indicate that that trait is present. Okay? So here we have some organisms. Here we have a lancelet, which you'll learn about later, lamprey, bass, frog, turtle, and leopard. And then we have traits like backbone, hinged jaws, limbs, forelimbs, amnion, and hair. And you can see the lancelets have a 0 for this trait here. They do not have hinged jaws. They do not have limbs. They do not have an amnion. They do not have hair. This is our outgroup. It's a group known from the fossil record and from other places to diverge first. So it helps us to figure out the order of evolution. So lancelets are the outgroup. Then when we go to lampreys, lampreys do have vertebrae, okay, but they don't have the other traits. And bass, for example, they do have vertebrae and they have jaws, but they don't have forelimbs, so on and so forth. You can see how this is put together, and you can use this information to draw a tree and then to label where the ancestor likely was that first evolved these traits and how it passed them on to its descendants. Okay? It's a way for you to do that. So for example, everyone from here, everyone here is a vertebrate, right? And everyone here is an amniote. Everyone here is a mammal, right? So you can see the clades nesting within the other clades on the cladogram. It's a nice way to visualize this. This is one of the ways we build them. Now, this is how we do it with physical traits, of course. With DNA, it's somewhat similar. But I want to point out that we always try to follow the idea of parsimony when we build these trees. That means the fewest number of changes possible are depicted on the tree. The fewest number of changes. What do I mean by that? Well, we could, for example, let's say we're not following the idea of parsimony. We could say, well, maybe vertebrae evolved here, and vertebrae evolves here, and here, and here, and here. Well, that's not parsimonious. The most parsimonious place to put vertebrae, we know all these critters have vertebrae, but it's more likely that it evolved once and was passed on to all of the descendants rather than it evolved convergently lots of different times. Okay? Now, it's possible you will have some things that do evolve more than once, but on our first attempt to draw these trees, we assume parsimony and then look to see if that makes sense. The fewest number of changes. It's the simplest solution is the best solution, is the idea of parsimony. I also want to point out that in some kinds of trees that you'll see in different publications, branch lengths represent how much genetic change there is between one group to another. But you won't see that in, we're not going to draw trees like that in class or anything. But you will see variations in what branch lengths mean in some trees. Okay? Branch lengths also sometimes indicate time, and usually they'll have on the X or Y axis, however it's organized, you'll see that scale present. Sometimes we'll draw trees in lab that have a time as one of the variables, but now, just to rehash this idea then, when we draw these trees, we're usually using lots of data in real life, and we try to apply the idea of maximum parsimony, meaning the fewest number of changes is best. Fewest number of changes is best. So once again, we could have humans, and let's draw a more complicated tree here. That looks like crap, but here we go. And here's our outgroup. Let's say this is like dogs and cats and humans and chimps, right? Well, it could be that dogs evolve fur, cats evolve fur, humans evolved hair, which is basically fur, and chimps evolve fur separately, and fish or whatever do not have it. That's one way to look at it, but the most parsimonious way of drawing this tree would be to say that, well, the common ancestor down here to all of the mammals evolved fur and passed it on to all those descendants. Okay, so that's parsimony, and we want to keep that in mind when we're drawing our cladograms. I want to point out that using these techniques, we have determined that life falls into 3 major domains: domain eukarya, domain archaea, domain bacteria. You can see common ancestor of all life down here. Life is thought to have evolved one time on the planet and then diversified. We're all related to one another. The first eukaryote is that blue ball. The first life form is that one down there, for example. And then, of course, the kingdoms you're used to, animal, plants, and fungi, are shown in red here. And then there are other groups that have been classified in different ways at various times. And if we go back far enough, we find out that it's complicated to figure out how we're all related, because way back in the day, we had something known as horizontal gene transfer going on. You can read about that in the chapter where, for example, chloroplasts show up and they cross from bacteria into plants, and mitochondria were a kind of bacteria that's found in plants and animals and fungi. And back in the day, DNA sometimes crossed between unrelated groups because of viral infections or one organism living inside of another. And it makes some of this stuff kind of difficult to figure out because DNA is kind of all mixed up there in some cases, and it takes a lot of effort and a lot of research and a lot of creativity to untangle the web of life to figure out how we are all related. Okay, so in the last video that I'll do, we'll work through how to make a cladogram using the example from the end of your textbook. And I recommend you do this example, go to the end of the chapter and do this, and then come back and watch the video.